Hello, everyone, and welcome back to the Anatomy and Physiology podcast. We're going to be talking about the nervous system today. So first, let's start off with what does the nervous system do for the human body? What are the major functions of the nervous system? Go ahead and pause the podcast, see if you can name these and describe them on your own. But basically, the nervous system, it's one of its major jobs is to maintain homeostasis in the body. So if you can recall when we were talking about feedback systems and homeostasis, and really throughout the whole course, we've been talking about how our organ systems can help us maintain homeostasis. For example, when we get cold, our body shivers to warm us back up, or when we get too hot, we sweat to cool ourselves back down. And in each one of those systems, we tried to highlight and name a control center that's going to be monitoring different variables and coordinating different responses to help us maintain homeostasis. Most of the time, those control centers can be found in the brain, and they're going to be monitoring those variables and helping us maintain homeostasis. So one of the major functions of the nervous system is going to be part of all of those homeostatic systems that we've been talking about. Next, there's sort of three major parts or three major big pieces that the nervous system play a role in. First, it can receive sensory input and give us information about our surrounding environment. This includes internal stimuli like our internal body temperature, but also external stimuli like the ambient temperature in the room. So one of the functions of the nervous system is it can receive sensory input. Another function of the nervous system is it can send information out to muscles and glands and other effector organs. So when we receive sensory input, like that cold temperature hitting our body, it might start to drop our internal body temperature, and one of our responses might be to send information out to our skeletal muscles to contract and relax really fast or to shiver and to help us warm back up. So it can not only receive sensory information, it can also send out motor information. And then in between those two, our central nervous system and our nervous system in general has the ability to integrate information. So the brain and spinal cord can process the inputs from those sensory receptors, sending information towards our brain. And if needed, it can then transmit information to those motor neurons, sending information out to different effectors in our body. So those kind of three pieces fit together nicely. We can receive information from our environment, we can process that information, and we can send information out to our motor, through motor neurons to effector organs. And then finally, the nervous system plays this interesting role of allowing for our highest order mental activities and thought processes. These are things like consciousness, our ability to be aware of our own existence and aware of even our own thoughts. So all of those thoughts, memories, emotions, logical calculations that we need to make, all of that can only be done with the nervous system. Specifically, we're talking about the brain and its ability to process information. A couple major pieces of the nervous system. When we talk about the nervous system, we're going to go through a couple major things here. So let's just do an overview of some of the important structures and important pieces of the nervous system. First, our nervous system has two major tissue types, or two major, let's say, cell types that allow our organs of the nervous system to function properly. See if you can think of these two major cell types and describe them. They are going to be neurons that have the ability to send an electric signal across the cell, and glial cells, which are the supportive cells of the nervous system. Those cell types then come together to form all the tissues that make up the brain, the spinal cord, and all of the nerves that come off of and go into the brain and spinal cord. Speaking of which, our body has these long cords that they come off of the brain and spinal cord, or they go into the brain and spinal cord, and these things are called nerves. One of the major structures of the nervous system are going to be these things called nerves. And these are like bundles of axons that the axon of the neuron can travel out into the body or start in the body and then move into the central nervous system. And these nerves take two major forms. There are cranial nerves and there are spinal nerves. And let's think about the differences between these two things. Cranial means brain, and so these are going to be the ones that come off of and go directly into the brain. And then there are spinal nerves that come off of the spinal cord and go out into the body. There are 12 pairs of cranial nerves that I'd like us to know, and then there are 31 pairs of spinal nerves. And these nerves, they form a few interesting structures and they have a few interesting structures on them. One thing that we might notice about a nerve that's exiting the spinal cord and going out into the body is that there's occasionally a little bump on that nerve, a little bulge in that nerve. And we call this bulge, when it's out in the peripheral nervous system, when it's out in the body, we would call that a ganglion. And there's some regular repeated places that we can look for and see these bulges on the nerves and know that these things are ganglions. And those locations, one of them is straight off of the spinal cord, right on the dorsal root of the spinal nerve, and that's called the dorsal root ganglion, and there's a few other places that we can find these things. These bulges in the nerves, called ganglions, are where the cell bodies of the neurons of the nervous system, where they live when they're out in the peripheral nervous system. So when we have a neuron, not in the brain or spinal cord, but out in the body somewhere, we need to create a little bit of space for the largest part of the cell, and that little space that we would need looks like a little bulge on the nerve, and we would call that bulge a ganglion. Another important structure that happens with these nerves is called a plexus. A plexus. And this is like a network of axons, almost like a web of a bunch of nerves that kind of converge and move away from each other, forming this little web-like nervous structure. And there's 4 or so major areas where this can be found. There's a cervical plexus in the neck. There's a brachial plexus in the shoulder. There's a lumbar plexus just above the hip. And then there's a sacral plexus just below the hip. These plexuses are really great at allowing for information to jump between nerves, and then after the plexus, you'll notice that there's these cord-like nerves that then go out to their final destination in the body. All right, now let's talk about the major divisions of the nervous system. See if you can pause the podcast right now and see if you can name all of the breakdowns of the nervous system. First, we have generally the nervous system is broken into 2 major parts, the central nervous system and the peripheral nervous system. The central nervous system is the structures that live right on the midline or straight up and down the center axis of the body, and this is the brain and the spinal cord. Those are the 2 major structures of the central nervous system. The peripheral nervous system are all of the nerves and sensory receptors that exit the brain or spinal cord or go into the brain and spinal cord. And because we have 2 major types of nerves in the peripheral nervous system, one's going towards the central nervous system and one's going away from the central nervous system, we can categorize the peripheral nervous system into 2 major categories. We can have the sensory division of the peripheral nervous system or the motor division of the peripheral nervous system. And the sensory division are going to be the neurons that pick up information from our environment or even our internal conditions and send them towards the central nervous system. Typically, in the sensory division, there's also these things called sensory receptors, which are at the very ends of these sensory neurons, and they are kind of specialized cells that can pick up specific information, like photoreceptors might be a specialized receptor that can pick up light, and then they would then transmit that information to a neuron a sensory neuron that would transmit the information towards the central nervous system. So that is the sensory or afferent division of the nervous system. If we send information the opposite way, what if we start with the information in the brain and spinal cord and we want to send that information out to our muscles or glands? We would use the motor or efferent division of the peripheral nervous system. And this is where we send action potentials from the brain and spinal cord out to effectors like our muscles and glands. And in this division, we could send information to a couple different things. We could send information to skeletal muscles that are under our voluntary control, or we could send information to things that are under our involuntary control, like smooth muscle, cardiac muscle, glands, other organs. And the motor division is broken down into two major categories. The motor division is broken down into the somatic nervous system and the autonomic nervous system. And remember, soma means body, and the somatic nervous system is we're sending information out to skeletal muscles of the body. All of those big muscles that are connected to our bones, those are going to be the effectors of the somatic nervous system. And the cool part about the somatic nervous system is we're sending information to things that are under our voluntary control. And when we get to this point, we can talk about it has a single neuron chain. And so I'll clarify that in a little bit, but basically what that means is there's only one neuron that exits the spinal cord and goes out to our skeletal muscles, and so we would call that a single neuron system or a single neuron chain. And that's contrasted with the autonomic nervous system, which sends information out to things that are under involuntary control. These are going to be things like smooth muscle, cardiac muscle, and all of our glands that are under involuntary control. We don't really get to control whether our sweat glands secrete their sweat or not. That's going to be part of the autonomic nervous system, sending information out to those glands to dump their fluid. These, not only is the stuff in the autonomic nervous system under involuntary control, the neurons that send information out to those things look slightly different than the system used for the somatic nervous system. Where the somatic nervous system, the one that's under voluntary control, uses a single neuron system, the autonomic nervous system uses a two neuron system. And so we would see one neuron exiting out of the spinal cord, and it would synapse or connect with the second neuron in the system, and you'd see that cell body connecting to that first neuron, and then that second neuron, its axon would then go out to the eventual effector like smooth muscle, cardiac muscle, or glands. And because we can see that second neuron cell body out in the peripheral nervous system, these autonomic nervous system chains have a ganglion that we can see, a little bump on the nerve to create space for that second neuron's cell body. And it's in this autonomic nervous system that we have our three final subgroups here. So within the autonomic nervous system, sending information out to involuntary effectors, we can have the sympathetic nervous system, parasympathetic nervous system, and the enteric nervous system. The sympathetic nervous system is where we send information out to things that are under involuntary control, like smooth muscles. And in this division, in the sympathetic division, we'd be sending out information that's going to amp up the body and prepare it for physical activity. This one's oftentimes associated with stressful settings or fight or flight responses. Like if you narrowly miss an accident in your car, you have to swerve out of the way, your heart might start beating really fast, you might start sweating, your pupils might be really dilated and let a bunch of light in. Those are going to be all sympathetic responses. The parasympathetic nervous system, the second major category of the autonomic nervous system, is kind of does the opposite of the sympathetic nervous system. Its function is to help us go into a restful state and sometimes like a digestion and excretion state. So we'd want to kind of wait for the parasympathetic nervous system to turn on until we're in a really safe, comfortable location, ready to relax and chill out. Our parasympathetic nervous system would kick in and allow us to start digesting our food. It would allow us to calm down, possibly fall asleep, possibly even excrete some of the waste. That would be a good time to do that. That's the parasympathetic nervous system. More rest and a little bit of digestion. And then finally, the third breakdown of the autonomic nervous system is kind of the newest one. I'm going to talk the least about this one. It is the enteric nervous system. This one's sometimes just brought into and covered at the same time as the parasympathetic nervous system, but it's slightly different. This one, the enteric nervous system, is completely contained within the digestive system, and it has this kind of plexus, so this web of neurons, web of nerves that can spread out and can regulate the speed with which things move through the GI tract and the amount of gastric secretions that get made, things like that. So it's mostly just digestion. And that one is sort of controlled along with the parasympathetic nervous system, but it can be on its own. So I'd like us to be able to understand and describe the differences between those divisions and specifically talk about its major functions. Okay, next up, let's talk about neurons and glial cells, the two major cell types of our nervous system. Neurons are kind of the main character of the story. These are the ones that can create an electrical signal and transmit it all the way across the cell. That's pretty cool because it's really the only cell type in the body, one of the only cell types in the body, that can create an electrical signal and send that electrical signal from one end of the cell to the other end of the cell. In addition to that, these neurons can be very long, so being able to transmit information from one part of the cell to another part of the cell means that we might be able to just send information all the way down our arm with one cell doing that. So this is pretty cool, and they're really sort of the main character of the nervous system. The neurons have three major parts and then a few other interesting structures that we're going to talk about. We probably already talked about the three major parts of the neuron, which are the cell body, or the main biggest part of the neuron, where the nucleus is held and a bunch of other organelles, maybe some mitochondria there producing a bunch of ATP. Then, around that central cell body, we have our two other really important parts. We've got dendrites and axons. Dendrites are going to be the part of a cell that picks up information and transmits that information towards the cell body. And so this might pick up information like in our skin and transmit information towards the cell body, which we're then going to send to the central nervous system, or it's picking up maybe information from different neurons, culminating in sort of consolidating that information and then sending it out through a long process on one side of the cell, which is called the axon. So the dendrites are the parts of the cell that pick up information and send it towards the cell body. The axon is the one or multiple long projections off of the cell body that sends information away from the cell body. The other really important terms here and structures on the neuron that we should now know about are things like the axon hillock. So when the information is being consolidated in the cell body, it needs to travel through this funnel-like structure in order to get to the axon. And this funnel-like structure in between the cell body and the axon is called the axon hillock, and it is where we need to make enough charge that will then be transmitted down the axon. So it's kind of like where the charge distills down or kind of funnels down towards the axon, and we need enough charge to build up there in order for the charge to be sent down the axon. We've got things like myelin sheaths that can be wrapped around the axon that will protect the axon and increase the speed of transmission of the axon. In between those myelin sheaths are these little gaps in the myelin sheath where we can see the exposed axon underneath it, and those little gaps are called the nodes of Ranvier, and that is where we can increase the speed of transmission or allow the signal to sort of skip across the axon and move really fast. And then finally, at the very end of the axon, the axon sort of splits up into multiple smaller projections, and we call these things presynaptic terminals, or sometimes they're called axon terminals. And it's going to be those things that are going to synapse with or connect to a different structure that we're trying to get this information to. Those structures that the axon terminals connect to could be things like other neurons, that's super common, or things like muscles or glands that we're going to try to stimulate. And when we think about neurons, there are two major ways that we can classify neurons. See if you can pause the podcast and describe these two major classifications of neurons. I'll give you a hint if you don't know. There's a functional classification of neurons and a structural classification of neurons. Just like when we talked about joints, joints could be described based on the function that they have, how much range of motion they offer, or they could be described based on their structure, what are they made up of. Neurons are the same thing. So we can have a functional description of neurons, and the functional classifications are sensory or afferent neurons, motor or efferent neurons, and then interneurons. And basically these just describe what the neuron can do. Sensory or afferent neurons pick up information and transmit information towards the central nervous system. Interneurons are inside of the central nervous system, and they process the information, and if needed, they transmit information to the last type of neurons, the motor or efferent neurons, which transmit action potentials away from the central nervous system to effector organs. Structurally, though, we'll notice that some neurons look very different from others. The typical neuron that we always look at and use to study the nervous system are called multipolar neurons. It's the neuron that we drew in our notes. It's the neuron that we typically see in diagrams. It's got one cell body with multiple projections coming off of it, and those are dendrites, and then one really long axon moving information away from the cell body, the classic neuron shape. This is most of the neurons in the central nervous system, to be honest, and even the motor neurons that take information away from the central nervous system look like these multipolar neurons, and so that's why we mostly just talk about these ones. But there's two other structural classifications of neurons that I'd like us to be aware of and not be too unfamiliar with when we see neurons drawn in this way. The other types of neurons are bipolar neurons and pseudo-unipolar neurons. Bipolar neurons have their cell body right in the middle of the cell and have two long singular projections on either side that then branch out into their dendrites and axon terminals. So it sort of looks like a center piece, a center cell body with two long projections on either side that then kind of flare out into their tree-like projections. And those are bipolar neurons. The best place and the place that we'll oftentimes find these things is the photoreceptors in the eye. We'd have these bipolar neurons that can pick up light information and then transmit it directly to a sensory neuron. The last major type, so we had multipolar, bipolar. The last major type is called pseudo-unipolar, or sometimes called just unipolar. I'm cool with either one. Unipolar neurons have their cell bodies kind of off to the side, and the rest of the structure is one singular long axon dendrite connection. And so what I mean by that is there's one cell body off to the side, and the rest of the cell is a bunch of dendrites looking for sensory information in some part of the body, and then those dendrites form a straight line connection directly to and down an axon, and then that axon then synapses with the cell that we're trying to get information to. So it's kind of like we take the cell body and pretty much eliminate it, just throw it off to the side and allow for one unipolar directional movement, a dendrite connected directly to an axon. That's why we call it unipolar. And those ones are going to be found in mostly in the sensory division of the nervous system. So those are our neurons, the main characters of the story. But what about the sidekicks? What about the other cells that allow our nervous system to function properly? Well, these are going to be our glial cells. There are 6 glial cells that I'd like us to be able to identify, describe their function. In these 6 glial cells, go ahead and pause the podcast now and see if you can name each one of them, name their major function, and then describe where we would find these things in either the central nervous system or the peripheral nervous system. So here we go. The first one that we can cover are these things called astrocytes. Astrocytes are these star-shaped cells in the central nervous system, and what these guys do is the projections that come off of this astrocyte that looks like the sort of the arms of the star. Some of these projections touch our neurons in our brain and spinal cord, and other projections reach out and touch the blood vessels. And what's cool about these is they create a barrier between our bloodstream and our neurons or our brain. And this barrier between the blood and the brain is called the blood-brain barrier. And this allows us to keep our blood and our brain separate from one another, because sometimes when we ingest weird stuff, it can sneak right into the bloodstream, and we really don't want toxins or other foreign bodies to come in contact with our neurons of our brain and spinal cord, because the neurons in our brain and spinal cord, they do not regenerate if they're damaged. There's rare cases that they can, but for the most part, if we damage our neurons, they are damaged for the rest of our life. So our body has this like defense mechanism where we separate our blood from our brain via these glial cells called astrocytes. There are some things that cannot make it through the blood-brain barrier, and then there are some things that can make it across the blood-brain barrier. Most toxins, most chemicals don't make it across this blood-brain barrier,
And the things that do make it across the blood-brain barrier are like the serum or the plasma of our blood can cross there, which has got a bunch of water and ions and nutrients and sugar, all of that good stuff we want to just let right across the blood-brain barrier. There are, however, some weird things that can make it across the blood-brain barrier that maybe we wouldn't want to, but just happen to be able to move across the blood-brain barrier. This is things like alcohol. When we ingest alcohol, several minutes later, we start processing and absorbing that alcohol. It gets into our bloodstream, and that little alcohol chemical can travel straight across the astrocyte without the astrocyte even knowing it, and can affect neurological activity. A lot of other illicit drugs can do this. There are some pharmaceutical drugs that we want to do this. And sometimes in pharmaceutical research, our goal is to try to find the things that can move across the blood-brain barrier and use them in pharmaceuticals. Really common form of research. So those are astrocytes. If we had to make an analogy to what these things are, they'd sort of be like little bouncers or little bodyguards that are trying to keep stuff away from the neurons and keep them nice and healthy. The next major type of glial cell is these things are called ependymal cells. Ependymal cells. And these are going to be the cells that line the cavities of the brain and spinal cord. So given that they line the cavities of the brain and spinal cord, we're only going to find these in the central nervous system. And specifically, they form these little areas called choroid plexuses. And these cells can secrete and produce the cerebrospinal fluid in our brain and spinal cord. Some of it is just like filtering the plasma from blood, and some of it gets created directly from the ependymal cells. And so their main job is to create the fluid that the brain and spinal cord is bathed in and can slosh around in. And again, they kind of start at this area called the choroid plexuses. Their job is to make sure that all the neurons of the brain and spinal cord are comfortable, happy, have a bunch of nutrients, have no toxins around it. So they're sort of like they're keeping everybody happy in the brain and spinal cord. We also have these glial cells called microglia. Microglia are specialized immune cells of the central nervous system, and they're like macrophages that are looking for any type of ailments or disease or dead tissue or foreign bodies that need to be gobbled up and destroyed. And again, these things are called microglia. They're basically like little medics or little nurses inside of the central nervous system that are looking for things that shouldn't be there, invading substances that we want to eliminate. Microglia. Finally, the last glial cell of the central nervous system are these things called oligodendrocytes. Oligodendrocytes, oligo means several, and dendro means little projections, and cyte means cell. So these are cells that have a bunch of or several projections that come off of them, and at the ends of these projections, they wrap around the axons of the neurons in the central nervous system and produce the myelin sheaths in the neurons of the brain and spinal cord. Of the ones that need myelin sheaths around them, the oligodendrocytes create those myelin sheaths. And what do? They protect the axons, which are those thin tubes sending information away from the cell body, and they also speed up the rate of transmission in those cells. They make them transmit information a lot faster. So those are our four major glial cell types of the central nervous system: astrocytes, ependymal cells, microglia, and oligodendrocytes. Next, let's talk about the two major cell types of the peripheral nervous system. These are the glial cells that can be found out in the body, so not on the central nervous system. And these are going to be our satellite cells and Schwann cells. Schwann cells are analogous to those oligodendrocytes. They create the myelin sheaths that are out in the peripheral nervous system. So anytime we need to create those little myelin-coated axons in the peripheral nervous system, like out in the arms and legs and the body, we're going to recruit these things called Schwann cells to do that, and they speed up transmission and protect the axons. Satellite cells, they wrap around the cell bodies of the neurons out in the peripheral nervous system, and so they're trying to protect the cell bodies of those neurons in the peripheral nervous system. So where are we going to find these? Well, if they're cell bodies and they're out in the peripheral nervous system, we need to create a little room for these cell bodies to hang out in, and that little room is a little ganglion. So we're going to find most of these satellite cells in the ganglion out in the peripheral nervous system. And while we're here talking about myelinated sheaths and myelinated axons, let's now talk about what we know about and what is formed by these myelin sheaths. So when these myelin sheaths wrap around axons, not every neuron of the brain and spinal cord and peripheral nervous system has these myelin sheaths wrapped around them. Some do and some don't. The ones that have these myelin sheaths wrapped around them, we would call myelinated axons, and these are going to be the white matter of the brain and spinal cord. The ones that don't have these myelin sheaths wrapped around them, or at least they're kind of like hanging out, they got maybe a little bit of myelin sheath wrapped around part of it, these are going to be called unmyelinated axons, and we would find these axons in the gray matter of the brain and spinal cord. So these myelinated axons, the ones with myelin sheaths around them, they can transmit information really fast. Where one myelin sheath comes really close to the next myelin sheath along an axon, there's going to be these little gaps in the myelin sheath, and these gaps are called the nodes of Rempierre, and it allows for the signal to skip across the axon like a stone's being skipped across water, and it speeds up nervous transmission. Whereas unmyelinated neurons, these ones, they don't have the Schwann cells or oligodendrocytes wrapped around them. They're kind of hanging out next to them, and they get a little bit of protection from them, but they transmit their information way slower, and they're going to be in the gray matter in our brain and spinal cord. Speaking of which, if we look at the anatomy of the brain, we would notice that when we cut into the brain and look at the internal structure of the brain, we can see the different color of the tissue just with our naked eye. We can see some areas that are darker and some areas that are lighter, and it matches up to exactly what we just talked about, whether or not the neurons in that area are myelinated or unmyelinated. So when we have myelinated neurons, we would, with axons that have the myelin sheaths around them, that's going to be the white matter of the brain, and that's usually in the deeper part of the brain. Whereas the gray matter is more on the surface of the brain. It looks darker to our naked eye, and we can see, we would find unmyelinated axons there, and that's oftentimes called the cortex or the cerebral cortex of the brain. So on the cerebrum, the very surface is like a few millimeters to a few centimeters thick of gray matter that we would call the cerebral cortex. And I'll clarify here a few other main terms about the brain and spinal cord. So we talked about this a little bit, but when we're out in the peripheral nervous system, we're out in the main part of our body, we're sending information out to effector organs like muscles, or we're picking up sensory information and transmitting it towards the central nervous system, we have these long tubes that have a bunch of axons inside of them with connective tissue wrapped around the outside. Those long tubes, or those cords that are covered in connective tissue and have axons down the middle of them, when they're out in the peripheral nervous system, those are called nerves. And the nerves are named the regular repeated locations in the body. We can find the sciatic nerve and the ulnar nerve in our arm. So the sciatic nerve kind of down our hip and leg, the ulnar nerve up in our arm. And however, when we have those kind of cords of neurons, cords of axons in the central nervous system, in the brain and spinal cord, there's no connective tissue sheath wrapped around them, and they're kind of hard to even see where these areas are, but those are called nerve tracts. So nerves are out in our body, and they have a connective tissue wrapped around them, and we can sort of separate them, we can find them and name them, whereas the nerve tracts are going to be found in our central nervous system, and they're much harder to find exactly where they are because they don't have the connective tissue wrapped around them. In addition, we said that, cell bodies, when they're out in the peripheral nervous system, they need a little bump on the nerve, and we call that bump a ganglion. But when we're in the central nervous system, there's no bump on the nerve because there's no connective tissue coating around it. So instead what we get are just these groups of cell bodies in the brain and spinal cord, and when we have these areas of the brain and spinal cord that are densely clustered with neuron cell bodies there, we would call those things nuclei, or a single one of them would be one nucleus. And oftentimes we can name and identify those nuclei and they have specific functions in specific areas of our brain, and they sort of connect to different parts of our brain. So I'd like us to know the difference between nuclei and nerve tracts in our central nervous system, and ganglion and nerves in our peripheral nervous system. Okay, next up, let's start talking about how our body can store an electrical charge in our cells and transfer that information from one part of our body to another. And the way that we do this is through these things called action potentials. Action potentials are an electric charge that we can store inside the neurons or the cells of our nervous system. And it's pretty cool. They're one of the only cell types in the body that can do this, and it allows for a really fast transfer of information around the body. So it's really great that our body can do this. Otherwise, we would be relegated to just like chemically sending information around the body, which is really slow and tedious. So, how do we store this electrical signal in our body, and how do we transmit that signal from one end of the neuron to the other end of the neuron? Well, first, we need to store a charge inside of this neuron. And in order to do that, we can either pick a positive or a negative charge, and our body chooses to create a strong negative charge inside of these neurons. So the goal for our neurons is to create a strong negative charge and just hang out at that strong negative charge. And whenever we need to, we can send a positive charge along that neuron and send that information from one part of the body to another through a positive charge flowing through these negatively charged neurons. So how do we do that? Well, first we need to generate that strong negative charge. So what we do is we can pump ions inside and outside of our neuron and store up a negative charge inside the neuron. And the ions that we use here are sodium and potassium. And so what we can do is we can take our sodium potassium pumps, give those pumps some ATP, and spend that cellular currency, we spend that energy in order to pump sodium and potassium inside and outside the body. And what we do is, for every ATP that we give one of these sodium potassium pumps, we pump 3 sodiums to the outside of the cell, and then we grab 2 potassiums and pump those to the inside of the cell. And if we keep that process up, pumping 3 sodiums out for every 2 potassiums that we pump in, eventually we get a strong negative charge inside the cell. You might be asking yourself, why is that? Why would we get a negative charge by pumping positively charged sodium and positively charged potassium inside and outside the cell? Well, if you can recall, we're pumping 3 sodiums out for every 2 potassium that we pump in. So over time, we would store up a little bit of a negative charge. The rest of the negative charge comes from the natural negative charges of like the organelles inside the cell and the proteins that hang out inside the cell. So there's a little bit of a negative charge inside the cell already, and then this process of pumping these ions inside and outside the cell really stores up a strong negative charge. If we stick a voltmeter, if we stick a volt reader in our neurons, we can actually measure the charge differential from the inside and outside the cell, and we get about a negative 70 millivolts inside the cell. So pretty strong negative charge inside the cell after this process happens. If you're having trouble remembering where these ions are pumped to and where they hang out in, at the resting state, you can think of a neuron as being like a salty banana. A salty banana. So if we had a banana in front of us and I poured salt all over the surface of it, we would have a bunch of sodium on the outside of the banana and a bunch of potassium on the inside of the banana, because bananas have tons of potassium in it. And that's what a neuron wants to maintain at its resting state. When a neuron is just hanging out, ready to fire its charge, it's like a salty banana. It's got a strong negative charge, and it's got a bunch of potassium on the inside, and a bunch of sodium waiting on the outside. And now that we have those ions hanging out and we have this negative charge, now we can send a positive signal all the way down the cell. And here's how that works. When we stimulate a neuron, we open up small sodium channels and allow little bits of sodium to flood into the cell and start to increase the charge of that neuron. And if the charge inside of a neuron goes from negative 70 up to negative 55, if we continually increase the charge inside of the cell, at some point when it reaches about negative 55, it causes the action potential to be sent all the way down the axon. So we're starting to store up we store a strong negative charge inside of the cell. And then at the cell body, if we start to stimulate this neuron a little bit at a time, little bits of sodium start to flood into the cell, and it increases the charge slightly. And it's only when a certain threshold is met that the cell fires all of its information and sends that positive signal all the way down the axon. And that threshold that we need to meet is about negative 55 millivolts. If we stimulate a neuron enough, the entire charge floods down the axon hillock and then flows all the way down the axon. So, once we hit that threshold of about negative 55, then a few major processes take place. First, if we take a little sliver of that axon and look at what's happening as a positive charge flows all the way down the axon, first, we would go from that strong negative charge and we would depolarize the membrane and raise it up to a really strong positive charge in that area. And what we do here is we open up a bunch of those sodium channels and allow all of that sodium that we pump to the outside to flood into the cell and create a positive charge in that area of the axon. What happens then is that those positively charged ions flooding in, they start to move down the axon a little bit, and they cause the sodium channels in that area to open up, and we get a strong positive charge in that area. And that process happens all the way down the axon, a strong positive charge flooding all the way down the axon. Once that positive charge has been established in part of the axon, then the neuron has done its job. We're transmitting that positive charge down the axon. So after that, we're sitting at the strong positive charge, and we need to reestablish a negative charge so that the neuron can fire again. So after depolarization of the membrane, we need to repolarize the membrane. We want to get that positive charge out of this area and establish a strong negative charge again. And the first step in repolarization, we have a bunch of sodium ions inside the cell and a bunch of potassium ions that we already stored in there. So what we can do is we can open up a bunch of potassium channels and allow that potassium to flood out, and that's going to slowly lower our charge back down, all the way down from positive, about maybe positive 30, all the way down to 0, back down to negative 55, back down to negative 70, and sometimes it can even drop below that. It can get to negative 80, negative 90, negative 100. So now we have a really strong negative charge inside the cell, which is great. We do want a strong negative charge inside the cell, but at this point, something's up. Something's wrong here. We have a strong negative charge, but if we go to stimulate this neuron again, it will not fire. Because in order to stimulate the neuron, we would open up sodium channels and allow sodium to flow into the cell. But all of the sodium is still in the cell. Right now we have a bunch of sodium on the inside and a bunch of potassium on the outside. So what we need to do is we need to flip this. We need to get a strong negative charge still, but we need to pump all of the sodium out of the cell and all the potassium inside the cell. We need to get our cell back to that salty banana state. Salty banana, sodium on the outside, potassium on the inside. So, in order to do that, we need to activate our sodium-potassium pumps again to help us reestablish this resting potential. And this state of having a strong negative charge, but the ions being in the wrong spot, this state is called hyperpolarization. In a hyperpolarized state, we have the strong negative charge, maybe even more of a strong negative charge than we need, negative 80, negative 100. But the sodium potassium are in the wrong place. And I got to be honest, this is actually a brilliant way for neurons to fire. Because if you're imagining this positive charge flowing down the axon, the axon, as the charge is flowing down and we're releasing our potassium outside, it actually prevents the signal from working backwards. So when these sodium channels open up and sodium floods in, if some of the sodium moves backwards, it's not a big deal because it's not going to send the signal backwards. So this hyperpolarized state it is really important, is a really important step in the firing of an action potential because it prevents the backward flow of the signal back towards the cell body. It only allows the signal to move in one direction down the axon, which is from the cell body towards those axon terminals. So in order to get out of this hyperpolarized state, we need to activate our sodium-potassium pumps. And we spend some ATP, pump 3 sodiums from the inside to the outside, and grab 2 potassiums from the outside and pump them to the inside. And then we get back to our salty banana state of having a bunch of sodium on the outside, a bunch of potassium on the inside, and a strong negative charge that is ready to fire again. So I know that was quite a bit of physiology there, but I'd like us to be able to describe how that action potential can be transmitted across a small portion of the axon, hitting on those major steps there: the resting state, the depolarization, the repolarization, the hyperpolarization, and then the return to the resting state. So once this information has flowed all the way down the axon, and towards the end of the axon we have these axon terminals, how do we then get information from that one neuron? How do we get it to the final destination, maybe another neuron or a muscle or a gland? And what we get here are these things called synapses. And synapses are the connection between one neuron and another neuron, or one neuron and a muscle, or one neuron and a gland. It's basically a connection between a neuron and something else. So we're all the way at the end of our axon, at those axon terminals, and they're going to come really close to the neck structure, but there's going to be a small gap from one to the other. And this is where the neurological synapse is. Okay, so these synapses work like this. It's essentially, when we're talking about the nervous system synapses, let's talk about the junction between 2 neurons. This is the site with which one neuron can transfer its action potential to another neuron. And in this way, we have a presynaptic neuron, the neuron that is sending the signal, and a postsynaptic neuron, the one that is receiving the signal. There are 2 major classes, or 2 major types of synapses: electrical synapses and chemical synapses. And we'll talk about each one of these. And if you want, you could pause the video or pause the podcast and see if you can describe each one of these on your own. Basically, electrical synapses, these ones are less common than chemical synapses, and they are they more common? They might be more common. These ones might be, I don't remember. So, anyways, we have these electrical synapses, and they allow for the direct transfer of an action potential from one neuron to another neuron. And they do this because the axon terminals and the dendrites meet up together and have gap junctions in between them. And these gap junctions are these little channel proteins that span one cell membrane to another cell membrane, and they allow for the positively charged signal that's flowing down one axon to just easily transfer directly into the next neuron. And this is really straightforward. It allows for direct communication from one cell to the next cell. It's great because it's very fast, and there's not a lot of thought that's needed. If one neuron fires, the other one is going to fire directly. This might be like the connections between one motor neuron and another motor neuron, where the information just needs to directly transfer from one to the other. Chemical synapses, on the other hand, these ones have a lot more, a little bit more complexity here. In a chemical synapse, there's no direct connection between one neuron and the next neuron. Instead, there's a tiny little gap in between them where we need to take that electrical signal, convert it into a chemical signal, secrete the chemical across that little gap, and then the postsynaptic neuron, the one on the other side receiving the signal, can then detect the presence of these neurotransmitters or the presence of the chemical, and then fire their action potential if they want to. So if you remember the neuromuscular junction, it's very similar, where we would have a presynaptic neuron with some synaptic vesicles with neurotransmitter inside of it. And when that action potential comes down the axon and hits that axon terminal, it causes the synaptic vesicles to bind and merge with the membrane of that presynaptic neuron and dump its contents out into the small gap in between neuron 1 and neuron 2, the presynaptic neuron to the postsynaptic neuron. On the postsynaptic neuron, we're going to have these little receptors that can detect the presence of the neurotransmitter. And if the correct neurotransmitter hits the correct receptor, then it will change something about the postsynaptic neuron. It might just cause the postsynaptic neuron to fire directly, or it might prevent that neuron from firing, or it might increase the likelihood or decrease the likelihood of them to fire. There's actually a few different situations that could occur depending on the neurotransmitter and what the receptor does. So electrical synapses and chemical synapses are similar in that they transfer information from one neuron to another neuron, but they're different in the ways that they can do this. The electrical synapse is just a direct connection between two cells. It's basically like one neuron merges directly with the next one and they become basically the same neuron. The chemical synapse is more nuanced. It can release different chemicals, and it allows for subtle differences of signals. We could release a lot of neurotransmitter or a little bit of neurotransmitter, or we could release one neurotransmitter over another, and they might have different functions. And in that way, the analogy that I like to use here is electrical synapses might be like a rule in your email where every time you get an email from a certain source, it just gets auto-filtered into one of your folders. So if you always get an email from like spam, it might just automatically throw it right into the spam folder. No thought process needed. When one stimulus happens, it just completely puts it into a category. Whereas with chemical synapses, it might be like more nuanced rules to your email. It would allow for certain messages at certain times to be categorized in one way, and then certain messages in other ways to be categorized in a different way. Or it might be like you can communicate with a text. So instead of just replying to your text, you can put different messages in it, different images, and cause different responses. There's a lot more nuance to the chemical synapse. Okay, these receptors in our synapse, they bind to the neurotransmitters that are released into the synapse. And the neurotransmitters have a variety of different functions. And two major categories of neurotransmitters aren't the only two, but two major categories of neurotransmitters are excitatory neurotransmitters and inhibitory neurotransmitters. And the excitatory neurotransmitters, when they bind to that postsynaptic membrane, they excite that neuron and increase the likelihood of that neuron firing. Whereas inhibitory neurotransmitters, when they bind to the postsynaptic membrane, they decrease the likelihood of that neuron firing. Some common neurotransmitters that you're probably familiar with, we've talked about acetylcholine before. That is like the vanilla of neurotransmitters. If any time we would need just a direct communication signal between two things, we can just use acetylcholine. It's a great neurotransmitter to transmit information. That might be between like a neuron and a muscle. That's where we might find acetylcholine. The other one that I'm interested in us talking about is GABA, which is an interesting neurotransmitter that can block the signal from being sent. So if we stimulate a neuron with GABA, release that neurotransmitter and bind to a neuron, it'll decrease the likelihood of that neuron firing again, or it might block the signal from being sent through that thing. We're going to talk about these two in particular. But there are a bunch of other ones that I'm sure you're familiar with, like dopamine, which is kind of a mood regulator, norepinephrine, which is more of like a stress and sympathetic nervous system neurotransmitter, serotonin, another mood regulator that is oftentimes involved in like sleep and appetite stimulant. So many neurotransmitters that have many different functions. Let's maybe in a little bit, let's talk about acetylcholine and GABA and think about how these things function. So as we were saying, these neurotransmitters fall into two major categories: excitatory and inhibitory neurotransmitters. Now let's think about what that means. Excitatory neurotransmitters, they increase the likelihood that the neuron is going to fire. So if we release an excitatory neurotransmitter and it synapses with and hits this neuron, future signals that hit that neuron need less action potential or less signal in order to stimulate that neuron. And here's how that works. When we release an excitatory neurotransmitter onto a neuron, we create what's called an excitatory postsynaptic potential, or an EPSP, excitatory postsynaptic potential. And what that does is it depolarizes the membrane slightly. And by depolarizing the membrane, remember our neurons are like salty bananas. We've got a strong negative charge on the inside and a bunch of sodium on the outside, a bunch of potassium on the inside. When we hit it with an excitatory postsynaptic potential, it depolarizes that negative 70 charge and brings it closer to the threshold needed for it to fire. And in doing so, less stimuli is needed to fire that neuron in the future. In the opposite way, an inhibitory neurotransmitter creates what's called an inhibitory postsynaptic potential, or an IPSP, an inhibitory postsynaptic potential. And what that does is it takes our salty banana that's sitting at a negative 70 millivolts and it drops it down away, it drops the charge down and away from the threshold, which makes that neuron harder to fire in the future. And this process is called hyperpolarizing the membrane. So instead of only needing, 15 or 20 millivolts to get up to that threshold, now we need 20, 25, 30 millivolts in order to increase it enough to hit that threshold of firing. And so you probably are familiar with drugs or other chemicals that create these excitatory and inhibitory postsynaptic potentials. Let's think about, this isn't exactly how all of these drugs work, but let's just imagine alcohol. Would you say that alcohol is a stimulant or a depressant? And if you're familiar, alcohol tends to be a depressant. It tends to slow down reaction times. It tends to cause long delays in thought processing or other motor skills. What kind of postsynaptic potential would alcohol create in the neurons in our brain? Well, they would create an inhibitory postsynaptic potential, meaning it's going to bring our neurons away from their threshold, hyperpolarizing the membrane and making it harder for those neurons to fire. So you might go up to your friend who's had a few drinks at the bar or something, if you're over 21, and you might say, hey, let's get out of here. And they take a long time looking at you, processing that information, and they say, no, I don't want to leave yet. That long delay in reaction time could be due to this inhibitory postsynaptic potential. It just takes a lot longer and more stimuli for those muscles to fire. Or even worse, let's say that they're driving home from the bar and they're smashed, and they then are driving through an intersection and the light changes to red. And the amount of time for their neurons to interpret that red signal might be so delayed that they're already through the intersection before they realize that the light turned red and they smash into somebody, obviously why we don't drink and drive. Excitatory postsynaptic potentials would be the opposite. I mean, imagine a drug that is a stimulant that causes an excitatory postsynaptic potential in the brain. Maybe something like methamphetamines that cause less stimuli needed for those neurons to fire. So any type of little stimulus, our neurons are on these hairpin triggers that just cause them to fire. You can even get drugs that can bring our neurons so close to that threshold, get such an excitatory postsynaptic potential that these neurons will fire automatically on their own without any stimulus. And those might be things like hallucinations, that you're seeing things or hearing things that aren't really there. Those could be excitatory postsynaptic potentials that do that. Okay, last but not least, let's now talk about our body's ability to, or chemicals' ability to mimic, inhibit, destroy, or somehow alter neurotransmitters in the body. So first, let's talk about two neurotransmitters that we're familiar with, acetylcholine and GABA. Under normal conditions, acetylcholine is an excitatory neurotransmitter. When it crosses that synaptic cleft and binds to the postsynaptic membrane, it causes a muscle to contract or a nervous system signal to be fired. So it sort of just allows for transfer of information from one cell to another. Whereas GABA is a major inhibitory neurotransmitter. Whereas if we put GABA on the surface of a neuron, it hyperpolarizes the membrane and makes it very difficult for that neuron to fire, and sometimes it will not fire at all. And so those are our two major neurotransmitters that we can talk about, like mimicking those neurotransmitters or inhibiting those neurotransmitters. And let's take these two and walk through a few important examples. What if we were to mimic acetylcholine or mimic GABA? Go ahead and pause the podcast and see if you can describe what would happen if we were to mimic acetylcholine. So we have a different chemical that gets released into the the synaptic cleft, a different neurotransmitter that's going to mimic acetylcholine. Well, it's going to bind to the same receptors that acetylcholine uses, and it's going to mimic or cause the same functions of acetylcholine. And this might be stimulation of muscles or stimulation of cognitive functions. These are going to be our typical stimulants. And one thing that's really common that mimics acetylcholine is nicotine. When people smoke or ingest nicotine in any form, maybe put a Zyn, I don't know if Zyns have nicotine, but they put a nicotine pouch in their lip and they get some of that nicotine. It's a stimulant in that it excites our neurons. It causes them to fire more easily and more quickly. And for some people, it's a highly addictive feeling of having our neurons sort of fire super easily. What if we were to mimic GABA? Well, remember, GABA are the ones that inhibit neurons from firing. So drugs or neurotransmitters that mimic GABA would have the same functions as GABA. It would cause sedation, paralysis, muscle relaxation. The common drug that mimics GABA, or as a GABA agonist, are these things called benzodiazepines. And these are really strong muscle relaxers or sedatives. And again, for some people, these become highly addictive. They get really used to walking around in this very hyperpolarized and inhibited state where it takes tons of stimulus to trigger your neurons. You might be asking yourself, why would someone enjoy that highly sedated state or that highly stimulated state? That's probably a better question for like a psychology or a pharmacology class, but just in general, some people's normal neurophysiology is it can create a stressful state, and by altering their physiology, it might help someone relax, and it might become pretty addictive to them. For example, if you're highly depressed and you aren't receiving enough stimulus in your brain, then maybe being on a stimulant might make you feel pretty good and could become pretty addictive. In the same way, let's say you're like stressed and anxious and you have all of these sounds around you that are bugging you all day, all these thoughts running through your brain. If you go on a sedative like a GABA agonist, it could cause a sense of relief in that those racing thoughts tend to calm down a little bit. Not certainly something that I would encourage us to do, using illicit drugs to, I guess nicotine is an illicit drug, but using drugs in general as a mechanism to deal with those symptoms, but certainly something that we're all going to be very familiar with. Okay, what about neurotransmitters that block or inhibit these neurotransmitters? So like we take an illicit drug or we ingest a chemical that blocks or inhibits any of either one of these two, acetylcholine or GABA. Well, if we block acetylcholine, it blocks the function of acetylcholine, which means that we might have reduced muscle activity or even paralysis. And what's a good example of a chemical that blocks acetylcholine? Well, I think we've talked about one before. Botox, the botulinum toxin, that's a good one. And then there's this other chemical called curare, which is another really intense sort of paralysis-inducing chemical. And those are going to be chemicals that inhibit acetylcholine. Well, what if we inhibited GABA? Well, then we would block the function of GABA. And the function of GABA is to block information from being sent around the brain into muscles. So if we were to, it's kind of a double negative, but if we were to block GABA, then we would cause unexpected continual transmission of a signal or stimulation of muscles. So we might see muscle seizures, really excessive excitation of um, of the neurons. This one, there's not a ton of medically relevant uh, chemicals that I can think of, but one of the most obvious ones is Narcan, which reverses the effect of like a massive sedative. So if you have somebody who's on a massive sedative or like an opioid and they've overdosed on the opioid, you can hit them with Narcan, which is a GABA antagonist, and it blocks this GABA function, and it will sort of reverse their massive overdose or the blocking of neurotransmitter. Other things that we should be aware of. I'd like us to know if we were to like bind to a receptor, well, then it would cause either one of those two neurotransmitter functions. If we were to destroy acetylcholine or GABA or any other neurotransmitter, we would obviously reduce the function of that neurotransmitter. And then if we were to introduce an enzyme to break down neurotransmitters, it'd have this a similar function as destroying them, but more of a short-term effect. By breaking them down, you can sort of reabsorb them and recreate the neurotransmitter pretty fast. And so I'd like us to be able to describe those differences in, or those different effects on neurotransmitters, mimicking neurotransmitters, inhibiting neurotransmitters, binding to receptors for neurotransmitters, destroying neurotransmitters, and degrading or breaking down a neurotransmitter. Okay, last but not least, let's talk about neuron regrowth, the ability of a neuron to regrow after damage. Most of the time, damage to neurons is permanent, and it once you damage a neuron, it will not recover. But there's a couple small instances where regeneration is possible, and here's the deal. Neurons that are in the peripheral nervous system seem to have a higher likelihood of regenerating over time. So if we got like a sensory or a motor neuron that is out in the peripheral nervous system, picking up information or transmitting it to muscles, it's possible that those ones could regenerate. Certainly, they have a higher likelihood than if they were in the central nervous system. If the damage to the neuron is pretty far away from the cell body, then it's a greater likelihood that this neuron can regenerate. If it's really close to the neuron cell body, it's less likelihood that it would break. And if it's right by the axon terminals, there's a greater likelihood that we could see some regeneration. As well as if the neuron is myelinated, so if they have those Schwann cells wrapped around them, because we're going to typically find this in the peripheral nervous system that they can regenerate. So if there are Schwann cells around them, there's a little bit higher likelihood. And if there's other immune cells around, like macrophages, we can typically see maybe a little bit more neuron regrowth. Still a lot of research needed in this area, but that seems to be the major instances where we see neuron regrowth. Okay, thanks for listening this far. If you've gotten here, good luck studying. I will see us all soon.